Scientists at Columbia University have just edited the DNA of early human embryos with a level of precision that has never been achieved before. The research, led by geneticist Dieter Egli, used a technique called base editing to correct individual genetic letters inside fertilized eggs and two-cell embryos donated by parents. I am Soumya Pillai, and in this episode of Pure Science, we will unpack the findings. For the study, which is currently in preprint, researchers from Columbia University successfully altered two genes, one linked to higher LDL levels and heart disease risk, another that controls hemoglobin production in fetuses. In some experiments, both genes were changed in the same embryo. The paper, titled Efficient Base Editing and Development in Human Embryos Without Chromosomal Alterations, found that unlike CRISPR, base editing did not cause chromosomal abnormalities or large deletions. Small errors were rare, and any off target activity, such as edits hitting the wrong part of the genome, depended on the specific guide the RNA used. This means it can potentially be controlled by careful design. To understand what these researchers did and why it matters, let's first try to understand what DNA actually is. DNA, or deoxyribonucleic acid, is the hereditary material in humans and almost all other organisms. It is like the instruction manuals for our bodies, and it contains the exact chemical codes needed to build, grow, repair, and operate, determining everything from our hair color to how our organs grow and function. Even a single alteration in this can cause severe conditions such as sickle cell disease, cystic fibrosis, or an inherited heart disease that can kill someone young and otherwise healthy. Now, what I'm telling you about are not lifestyle diseases. They are written into a person's code even before they are born. For years, the tool scientists turned to for fixing those errors were CRISPR-Cas9. It became a major scientific breakthrough after 2012. It can be understood as a molecular pair of scissors that could snip out a faulty section of the DNA and replace it. It even led to a US approved treatment for sickle cell anemia in 2023. But CRISPR is not precise enough for embryos. Sometimes it misses the target, sometimes it cuts the wrong material entirely. In 2020, the Colombian team, in an earlier version of the research, used CRISPR on embryos carrying a mutation in the EYS gene, which causes hereditary blindness. The hope was that each embryo would repair the faulty gene using its healthy copy as a guide. But that happened in only some embryos. In about half, the repair failed. Some lost long stretches of DNA, some destroyed the entire chromosome carrying the EYS gene. The team described the outcome as having absolutely catastrophic consequences. Base editing was built to avoid exactly this setback. Created in 2016 by Harvard geneticist David Liu and colleagues, it does not cut DNA at all. Instead, it makes a tiny nick in one strand of the double helix and guides the cell to fix the mutation itself. So it is less like cutting a page out of a book and more like correcting a typo. The Columbia team found none of the large-scale chromosomal damage that plagued earlier CRISPR experiments. That is the headline result, and it is genuinely significant. But the new method is also not perfect. Sometimes the base editing molecules fail to reach their target, which meant some cells in an embryo were edited while others were not. Scientists call these mosaic embryos, a genetic patchwork rather than one clean, uniform change across every cell. If a mosaic embryo developed into a baby, that unevenness could cause medical problems in ways that are difficult to predict. But the team is also not overselling the research and accepts that it will need more work before it can be a dependable medical procedure. Independent experts echoed that caution. Many clinicians and fertility experts have called this research promising, but said that the full results need to be scrutinized once published. Some even warned that safety checks would need to go beyond scanning for damaged chromosomes because it is possible that some of the potentially harmful effects would not be evident until after birth. That is the core anxiety that runs through all the embryo editing research. A change made before birth could shape a child's life in ways that do not show up in any lab test. There is also a practical dimension. This kind of editing could eventually help IVF patients implant embryos that would otherwise be discarded. But there is still a lot of work that needs to be done before the research progresses towards that conclusion. And a US based DNA testing company, Nucleus Genomics, has now even come forward to funding the next phase of this research, which will look into avoiding mosaic embryos and test base editing on embryos of around 100 cells. This is the stage at which fertility clinics typically freeze and examine embryos. But like I said before, the risks and the ethical concerns attached to this research cannot be ignored. One of the biggest concerns is that base editing could attract people looking not to fix inherited diseases, but to enhance traits. And this fear has history behind it. In 2018, Chinese scientist He Jiankui secretly created the world's first gene-edited babies. Twin girls whose embryos he altered to resist HIV, forging ethics approvals along the way. China jailed him for 3 years. The global scientific community was also united in its condemnation, not just for the act, but for the recklessness. But the Columbia research is nothing like that. It is careful, transparent, and openly presented for scrutiny. However, it is happening in the same field with the same tools, and it is pushing towards the same frontier. So, are engineered babies next? Well, not immediately. The science shows that embryo DNA can now be edited more precisely than before, but also that the method is not yet reliable enough for clinical use. That is all from me this week. I am Soumya Pillai, and you are watching Pure Science.